So, good morning everyone. So, I hope that everyone is safe at your home, and today we are going to start the second video of the chapter, that is the lesson, the cell, the basic unit of life. So, once we'll have a revision of the video 1, what we have learned. Understand? So, here we have learned that why the cell is the basic unit of life. We have learned the introduction, and here next we have learned some of the great scientists who contributed and their valuable observations regarding the cell, and who is the first person to observe the cell and the discovery of cell. Then we have seen about the Antony van Leeuwenhoek, and he is the first person to observe the living cells and made by his own magnifying lenses, that is called microscope. Then here we have learned that the simple, that is same experiment which the Robert Hooke did, we have did in observing the mastic. Yes or no? So here what we have learned, the Robert Hooke, what he said? He said that when he observed a bark, that is a soft bark of oak tree, when he took a thin slice and placed in a microscope which is prepared by himself, and he observed, what he observed? He observed it looks like a honeycomb, where he observed a small empty spaces, and he given the name as the little, that is cell. In Latin, cell means little room. Okay, Nana? So we'll have a revision again from the activity 1 to activity 2 and activity 3. So what are our topics? So our topics, today's topic, the topics should be covered in this session, means in this video. So what we are observing? So complete theoretical part in detail we will go. Observing the mastic. Observing the mastic, which I already said in previous, but I want to revise you. Then observing onion peel cell, observing the cheek cells. So here you are observing the plant cell and you are observing the animal cell. Understand? Observing the plant cells and animal cell. Then I will tell the difference between the plant cell and animal cell, which is not there in your textbook and very frequently asking in our exam regularly. Okay, Nana? Let's start. This is a similar experiment done by Robert Hooke. Here what we said, here we cannot take the bark of oak tree. So instead of that one, we took the matchstick. Instead of bark, we took the matchstick. Let us start our session with each and every experiment, and finally at last, definitely whatever I said in the video 1, I want to tell the parts of microscope. So let us start with the observation of the matchstick. So first aim. What is our aim? First one, aim. I am going to draw with a different chalk pieces. With that we can have. So aim. What is our aim? To observe the cells of matchstick. Understand? To observe the cells of matchstick. To observe the cells of matchstick. So instead of oak tree, we took bark of oak tree, we took with the mastic. Then apparatus. So what apparatus we require? So we require apparatus. We require matchstick. Then we require a cup of water where you have to soak the mastic for half an hour. Then we need a blade to cut the mastic thin slice. So it is used to cut the mastic a thin slice. We are taking the thin slice of mastic. Thin slice of mastic. Then here we are taking glass line. So everyone please watch. This is the glass slide. So this is the glass slide, Nana. Then you are taking cover slip. So I want to show you a cover slip. So this is the cover slip. Okay, this is, are you observing the cover slip? So very thin, you can see. Okay, cover slip. Then we need microscope. So these are the apparatus required. So in your textbook directly the procedure is given. Take the mastic, soak it for half an hour, then take a thin slice of the mastic, place it on the glass slide and cover with the cover slip and observe under the microscope. But here I am dividing everything in detail. Why? Because in your exam they will ask these type of experiments are very, very important for 4 marks. So children, you have to know what is our aim, what are the apparatus required. So here when you see in our summative 1 and summative 2, generally they will give the aim, they will ask to write the apparatus. So that is the reason I am differentiating everything very clearly whenever I am explaining the lesson itself. So what is our aim? To observe the cells of mastic. This is an experiment. Even he may give the question in that way. Here do the similar experiment like Robert Hooke. Understand? Similar experiment means the similar experiment which was done by Robert Hooke, the oak tree, the bark of oak tree. Here instead of bark of oak tree, we are taking the mastics to observe the cells of mastic. So the question will be given in this way. So these are the apparatus required. We need water. Why? Because to soak the mastic. Understand? We need a blade to cut the mastic, the thin slice of mastic. We are taking a thin slice. Why? Because if we soak them half an hour, easily the water is absorbed by the mastic and easily we can get a thin slice. Then you need a glass slide, we need a cover slip and we need a microscope. So these are the apparatus required for these experiments, for this experiment. Understand? Then next procedure, take the mastic. Then I hope that every children, that is who are watching this video, that everyone understood what is the aim and what is the apparatus. Why we have to write the aim and apparatus? By according to our examination pattern, they are asking differently. Means they may ask the question in not directly write about the experiment. They may ask apparatus, otherwise they will jumble the procedures and then ask them to put them in an order. So that is the reason every point from textbook is important. Please make a note on your mind that procedure. Whenever I will write the procedure, I am writing in a flowchart method where you people can easily catch the point and easily and can remember. So this is the procedure. First step, then after other step, then after other step. So get ready with the next procedure of how to observe the slide, that is cells of mastic. Okay, Nana? So let us see the procedure. So we will go procedure? Take matchstick. Take a matchstick. Okay. Soak it in water for half an hour. You can use 1 hour also. Understand? Soak it, then half an hour, and cut. Cut thin slice. Cut thin slice. Cut the thin slice of matchstick and place it on glass line and put a drop of water and cover with the cover slip. Here only already we have learned that when you are placing the cover slip, please keep in your mind that here no air bubbles should come. Ma'am, if air bubbles come, what we have to do? Just tap over it, then see under the microscope. Okay, then this is the first procedure. What is our procedure here? Then once again I want to repeat the procedure. Take a matchstick, soak it in water for half an hour. Here the time is very important. Understand? Then cut thin slice of matchstick, place it on glass slide, put a drop of power, cover with the cover slip and observe under the microscope. When you observe, you can see same whatever the structures which are given by the robot, same empty cavities only you will see. Why? Because the matchstick is already dead. So no, you cannot see the contents which are present in the like a living cell. So here we are going to do an experiment over the living cells, which we are taking a plant cell, that is onion bulb we are taking. Then we will take the cells over inside our cheeks. Okay, now get ready. We are going to see another experiment, that is other activity which is very important. What is the difference you are observing in the matchstick? If you see, the matchstick cells will be like this. Empty spaces. Okay, Nana? So we'll go for the next activity, that is the observing the onion peel. Observing the onion peel. So what is our first topic? Observing the matchstick. Then we are going to observe the onion peel cell. So let us have so next one is, what is our aim? Say, what is our aim? Aim indicates what you are going to observe. To observe the cells in onion peel. So to observe the cells in the onion peel, we need apparatus. So what are apparatus we require? We require onion bulb, then we require the blade or a knife to cut, then we need glass line, then we need cover slip, then we need a drop of water, along with we need glycerin. This glycerin is used not to dry the slide, that is material which is present in the slide. Material should not be dried. So then we need a brush to spread it uniformly. Then microscope. So these are the apparatus required for our to observe the cells in the onion peel. Now next, here what is our procedure? So let us see the procedure. First, take the onion, a piece of onion from the bulb. So this is the bulb of the onion. Understand? Then here I took a piece of the onion bulb. Then I broken down. I have broken them into two pieces. When you observe, you can see one translucent substance is attaches these two pieces. Are you observing now? One translucent substance, that is a translucent layer, we will say as a layer, a translucent layer which attaches the two pieces. Understand? This translucent layer we are taking and placing it on the slide. Okay, first what we have to do, just take the peel of onion, so to take the peel of onion, take a piece of onion bulb, then break it into two pieces, then you will get a translucent layer which attaches these two pieces. Cut this layer with the help of blade or scissors, then place it, cut it and place it over the slide, over the glass slide, and put a drop of water. And while you are placing this translucent layer, please keep in your mind, spread it uniformly. You have to spread it uniformly without any foldings. Without any foldings, you have to spread it uniformly with a brush. Then put a drop of water and glycerin. Why? Because glycerin will not allow the material to dry. Then cover it with cover slip. So here our preparation of the slide is completed. So here to take the peel of onion, place the onion bulb, break it into two pieces, then you will get a translucent layer. This translucent layer attaches to the two pieces. Then here when you cut it with the scissors or blade, then place it over the glass slide and put a drop of water. When you placing over the glass slide, please keep in your mind there should not be no foldings, it should be uniform. Then put a drop of water and add glycerin and cover with the cover slip, and then you can see under microscope. Then here I am showing this book to you everyone, which, see, are you observing the picture? This is the onion cells, the peel of onion cells, where you can observe, you can see, I want to draw some of the parts of these one. Okay, Nana? Just I hope that everyone is watching here. You can see, I will show you here. These are the onion. This is a slide. When you observe under the microscope, you can see these type of cells. Here you can see they are arranged in rows, and you can see middle dots are there, and here clearly you can observe a outer layer is clearly you can see visible. This is called as cell wall, which I am going to draw on the board. So everyone, I am going to draw on these are the diagram of onion peel cell. You can see clearly a dot in the middle, it is called nucleus. So I want to mention the part. Why? Because every time in the examination, the questions are asking, they are giving this diagram and asked to write, that is, please write the parts of this diagram. So this is the cell wall. You can see this is the cytoplasm and this is the nucleus. Why? Because we are going to learn about the nucleus in the upcoming classes. So here you can see mentioned clearly that you are seeing a well definite structure of the cells. Please listen my version. Well definite structures you can see, which you are not able to see these structures in next in animal, that is in cheek cells of human beings. Why? Because in human cells there is no cell wall. Understand? There is no cell wall. So these are the main parts, means these are the cells which I want to show you, that is through our aim is to observe the cells of the onion peel. So please, I hope everyone understood what I am saying here. These are the structures: cell wall, cytoplasm, nucleus. These are the structures which are present. If you see, there are various cell organelles which can be observed through the electron microscope. Okay, Nana? So here when you go for another activity, observing the human cheek cells. Let us see the third activity, activity 3. Activity 3. In activity 3, understand what we are observing. What is your aim? My aim to observe or observing cheek cells. Understand? We are observing the cheek cell. What are the apparatus required? So here, now everyone, here we need a toothpick. Otherwise we need wooden spoon to collect the materials from our cheeks. Either we can use spatula. Any of these three we can use. We need glass line. We need cover slip. Understand? We need glass line, cover slip, microscope, drop of water we need. So these are the things we need. So here when you see in this activity, some care we have to take while doing these experiment. Why? Because like onion peel, if we break it, we can get a translucent layer. But here we are collecting the cells from the cheek. Understand? So here while you are collecting the cells from your cheek, you have to be very careful. While before you are collecting, you have to wash your mouth thoroughly. Why? Because in our mouth there are thousands of bacteria will be there. So while you are collecting the cells over the cheeks, so here what we have to do, we have to wash our mouth thoroughly before collecting the cells from cheek. So this is the first one. While you are, then when you are taking the cells from cheek, just slightly scrap. Don't scrap hardly, otherwise your buccal mucosa will injure. That means inner layer. We are collecting the inner layer of the cells in our cheeks. Again I'm saying, we are collecting the inner layer of the cells in the cheek. So while collecting the cells in the cheek, we have to be very careful, otherwise there will be damage of inner layers of our cheek. The bleeding may occur. So first point, while doing a procedure, while collecting the cells from the cheek, we have to wash our mouth thoroughly, take the wooden spoon or a plastic spoon or a spatula, slowly collect the inner line. and collected material, place it on a glass slide. Understand? Then here when you place it, put a drop of water where it spreads uniformly. Then place the cover slip over it. See, once again I am telling, please take care. Under the guidance of someone only, you have to do this experiment. So, after collecting the scrap material, place on a glass slide, put a drop of water, then place a cover slip over it and observe under the microscope. So, once again I want to tell here, take two things in your mind. First, wash your mouth thoroughly. Second, while you are doing, that is, do not scrap too hard. Do not scrap too hard, which causes damage to the inner lining of your cheek. Then collected material, you have to place it on a glass slide, put a drop of water and spread it uniformly and place the cover slip over it and observe under the microscope. Here, when you observe under the microscope, the structures will be like this. So here there is no definite shape. Why? Because the cell wall is absent. Only cell membrane is present. And here the cell membrane is present. Where in plant cell wall is present. This is the major difference between a plant cell and animal cell. Understand? So every time in summative they are asking generally from these topics what is the experiment of observing the onion peel cell. First question. Second, what are the apparatus required for the onion peel cell? Third question, write the complete procedure. They will jumble everything, placing a slide down and washing the mouth. Third one, like that they will place all the steps unevenly and ask them to put them in an order. So every step from the lesson or from the textbook is important while you are reading. So everyone I am saying here, when you are taking the material, spread it uniformly, put a drop of water, cover the cover slip. If any bubbles, please tap them. The bubbles will go and you can see the picture, the cells clearly. When you observe the cells, you are seeing a lot of difference from onion cells and the cheek cells. Why? Because here they are not arranged in rows. There is no particular shape. Why? Because the cell wall is not present, which is present in the plant cell. Understand? I hope that everyone understand this topic very clearly. Why? Because every step is very important and diagrams also important. Here you can see there is an intracellular spaces. When you see there is a little bit, that means the compactly arranged. When you compare to the cells of plant within all, the lot of difference will be there. So if you see the cells of the cheek and the cells of the onion peel, a lot of variation is there. I hope everyone are you able to understand the topic. See, next time definitely I will show the experiment of these one with all the equipments. Okay, I hope everyone understood. Thank you so much for listening my video.